There's something that is very therapeutic about going out in the garden. What I love about those succulents is how weird and wild they are. They grow in an environment that is inhospitable to almost everything else. And I find those creatures, those plants, to be some of the most fascinating aspects of biology and of life. One of my graduate students brought a paper to me on artemisinin. It had been recently discovered, and shortly thereafter it was going to be recommended by the World Health Organization as the drug for treating malaria. I was fortunate to be able to go to Kisumu, which is on Lake Victoria. I got to go to a clinic, and outside the clinic were children lined up all the way around the clinic. Many of them were ill from malaria. It was just endemic. It was everywhere. And so this just ingrained in me how important this project was, because we had to deliver a therapy that was extremely high quality and extremely low cost and available all of the time. I want us to be very careful about tinkering with the planet. At the same time, we can engineer bacteria, we can put them into tanks in a carefully controlled environment and get them to produce life-saving drugs that might otherwise require a lot of intense farming or extraction from rare plants. After about a year and a half of working with the Gates Foundation, we finally had a grant. The Gates Foundation, they wanted to scale it up and get it out to people. So that's when we got the idea that, gosh, this is something that maybe we should start a company around. And that company is Amyris.
I joined Amyris several years after Jay had already wound down his direct involvement with the company. Jay is an absolute giant in the field of metabolic engineering and synthetic biology. Amyris' objective of making a specific molecule, artemisinin, more available to the people who needed it the most. That's actually fundamentally what we're also doing today. We're figuring out what are the molecules that have really spectacular beneficial properties from nature, and we're figuring out a way to make them accessible and scalable. If you think about it, all living organisms effortlessly perform intricate, complex chemistry. Plants convert carbon dioxide from the atmosphere into an astonishing variety of molecules, which enable them to grow and reproduce. The instructions for creating these molecules are encoded in each organism's DNA. We can now write DNA programs to make specific molecules. We then assemble the physical DNA and introduce it into living yeast cells. This enables us to control the cell's metabolism and tell the cell to produce specific molecules. Our yeast are living factories which efficiently convert simple building blocks, like sugar from sugarcane, into the molecules we want them to produce. So a great example of an ingredient that Amyris makes is bisabolol. Bisabolol is an ingredient that's used very frequently in personal care and beauty. It traditionally has been through harvest of either the candia tree or through the harvest of chamomile. So when we make bisabolol through yeast fermentation, it requires over 200 times less land than if we were to source it from its traditional source, the candia tree.
Most of the plants in the Cerrado region, they are endangered, especially because of the land conversion and the agriculture expansion. If we don't need to extract the leaves of candia tree to produce bisabolol, it means less impact on the natural populations. We still have to discover new plants, and with that, new compounds and new drugs could be developed based on the native plants in the Cerrado region.
I see glimpses of how we're going to use biotechnology in the future. It's really on us to learn and to invent how biotechnology can be used to impact these other fields.
here in the cutting edge. This time and this place is where all of these technologies are being developed, and they're changing. But we still need to drive the technology forward so that we can accomplish all of these things.
I ask myself, am I doing the right thing with my life? This is what I should be doing, because I think that this technology has so much potential to improve how we're making things and how we're sourcing things from the earth today. And I can think of no more urgent need.